Thank you once again for making it possible tonight. I'm delighted to have you all around. And so we are going to quickly begin the session so we can see how far we can cover up in the next 2 hours. Once again, you are all welcome. We just to do a recap of what we did on Tuesday. We had we had an introductory class where we looked at the modules and the essence of the training, what we'll be able to cover for the 1 month. And then at the same time, we now started the first module, which is Phytoinformatics. We opened us up into some of the databases that are used in phytoinformatics. So we did just one database. We used just one database. Can you see my screen?
Yes, I can see your screen.
All right, thank you. So we able to look at impacts. We're able to look at impact database, which is a database that has natural products created from about 4,010 medicinal plants in India. So if you are doing a work on phytomedicine, like I told us that you need about close to 1,000 or more natural products related to your plant. So in the assignment we did, my interest was just for we to be able to mine literatures, for we to be able to search literatures up to 50 literatures. That was actually my interest, was to mine up to 50 literatures, not just 50 literatures, but the ones that has informations on the bioactive constituents, on the bioactive compounds of the plant. Most of us did well. We tried. So I was not expecting us to look at database in that assignment. It was just to see some of the literatures that has been published on the chemical characterization of the plants, of the plant extract. So we did well. But I still want to repeat that part again, that even if you are doing any kind of research, you must mine literatures. It's very, very important. You must mine literatures because you need large number of bioactive compounds. So you need to mine as much as you can, as much as possible. That is the goal. You need to mine as much as possible. There are a lot of literatures that has information on the chemical constituent of your plant. So you learn to browse, search, use some of this database like PubMed, academia.eng. You try your normal Google search. So you just combine as much as possible to see that you have literatures that has to do with the bioactive compounds of your plant. Except your plant now is novel, that much work has not been done on it. That is a different case. That is a different case. But please, I will, for those who have not done it, who have not done assignment, or even those who have done something, but what you did was not really close, I appreciate you. Go back again and redo it, because you must have, first, you have a folder. You have a folder that contains all your 50, all the downloaded literatures. So all the literatures you will download, you keep them in a folder. Then you zip the folder. Then those literatures you have downloaded, you now check the bioactive compounds from the results, if they belong to the leaf or to the stem of that plant. You download the compounds using PubChem. When you download, if they all belong to the leaf, you cut them from your download and create a folder, call it leaf parts. You put them. If you open another journal that still has information on the leaf, after you download the structures from PubChem, you also put them back to that folder that is for leaf. Then if you open another journal that is talking about the stem that has bioactive compounds from the stem, you also download them, the structures one after the other in PubChem, cut them, create another folder named stem, put them inside there. So at the end, you may be having up to 2 or 3 folders. One for the general literatures you have downloaded, then the other folders for each of the plant parts and the downloaded structures. So it's a simple work. Then you zip it and send through mail. The essence is, if you want to start doing work on in silico macrognosy, then you must have your own database. You must create your own database. So I will be concluding in part today. I've said in parts, hope you can see my screen also.
Yes, sir.
All right. So in parts, we had 2 parts. Impact is divided into 2 parts. We have that of the phytochemical associations and that of the therapeutic use. So you can search by chemicals. So here we have already done this part where you click here and choose the plant you want to work on. So I want to explain something quickly from this part before I move to the therapeutic. So I can choose garlic. I can just choose to work on Garcinia cooler, or probably, let me use Morinda to explain. Morinda oleifera. Okay, there is no Morinda oleifera. Okay, so now I want to explain some little informations that you may get from Impact before we move to the other database. So today I will be explaining all the remaining databases we need for phytochemical. And this database search is very important because it forms the backbone of your research. So here when you click on each of those compounds, okay, I didn't mention this, but well, it will be necessary if you are creating a database that should be published, because those journals that you are downloading, okay, well, you are not creating a database to be published. If not, I would have said you take note of the citations of the journals you are downloading. But that will be necessary if you are creating a database that you want to publish for others to make use of. But it's not necessary now. Okay. So if you click on each of these compounds, like this one now, if you click on it, it will open up the dashboard that contains all necessary information you need about that compound. So with that, if you don't want to go to PubChem, you can still get some of the information here. This is a summary of the compound. Here you see some other database where this compound is found in PubChem CID, KMBL, in Sustain, KMBL, and others, and they are being presented with their code in those databases. So it also means that if you have a target of creating a database, then you link your own database with other databases that has similar information with yours. Then if you look down at their chemical identifiers, chemical identities, the SMILE code, the H3 code. So you can also copy the SMILE code from here to get further information you need, mostly in ADMET studies. We make use of the SMILE code. So you can get SMILE code from here. Yeah, so you can see the functional groups in that compound. So looking down, you just get any kind of information you need. Then you can get information on the physiochemical properties. So if you click on these physiochemical properties, I'm going to, in fact, ADMET, this impact has a lot of physiochemical properties associated to a bioactive compound. I don't think if there is any physiochemical properties you want to look for a plant that is not found here. Now, where this might be useful is maybe you have done a work with a library of compound, and at the end, out of 1,000 compound, you achieved 5 that we are better based on your studies. You can decide to look for informations about those 5. You can decide, okay, let me get their physiochemical properties. One, we quickly use Swiss ADMET or ADMET Lab. But then the physiochemical properties you will get in Swiss ADMET or in ADMET Lab cannot be compared to the one you can derive from this impact. You may begin to have a challenge that how will I type the compound directly in impact? You can search for compound directly. Although this one was gotten from the plant search, but you can see type in the name of the compound you want directly in this impact, and the impact will give you the information you need. So you can do search. You can search based on plants. That is what we are doing. You can also search based on the compound, based on your substance you have already. You can also search them here in impact. So I believe I'll quickly go to search in searching by compound. That will still lead us back to this page. So let's say you have a compound at heart, you have a compound in mind, and you want to do a finding about that compound. You can come down and choose here. Here is search by medicinal plant, but the other option is choose a phytochemical. So here you'll be the one to, you are now searching based on a phytochemical. You want to get the properties of a particular natural product. So you just need to click down and be patient. Sometimes it comes up, sometimes it doesn't come up. But once you just click, just be a bit patient. It will bring up a drop down box like before. Then you select the compound of your choice. So you can scroll down now to see if the compound you want is here, or you can search. Let's say I want camphorol. Maybe I did a research and camphorol happened to be one of my best hits, and I want to get information about this camphorol. before I can proceed. You can use PubChem also to get that information, but I will come to that when we are dealing on chemoinformatics. So we are still talking about the phytoinformatics. So you can use chemoinformatics tools to get chemical informations. You can also use some of the phytoinformatics tools to get natural product informations. So looking at this, there is no camphorol here. So I don't think if there is, okay, in this database, so there is no camphorol. So let me try quercetin. Quercetin, okay. So this is quercetin. If you click on it, then it will bring up quercetin. And one important thing is that you'll be able to see other plants that contains quercetin. Other plants that contains quercetin. So you can even use this to do a work of plant-derived quercetin. So you can, there are about 915 entries here that has quercetin, different plant species that has quercetin. So if you want to get information about that quercetin, you see all of them are all showing quercetin, the same code. So you just click on quercetin. Now it has come back to the page where we were before. So you can now get the physiochemical informations. So on your personal time, you have to study this and know the implications of this data when it comes to drug design. The molar weights, the log P, the TPSA, the HBA, number of hydrogen bond acceptors, number of hydrogen bond donors, number of carbon atoms, heavy atoms, hetero atoms, so stereochemical complexity, number of sp2 aldehydes. So there is a lot of information here that you can play around with. There are a lot. You can play around them. So you see a lot of information here on their physiochemical properties. Then you can also do a work on the drug likeness properties. So you can use that to do a work on drug likeness properties of quercetin. All right, are we together?
Yes, sir.
All right. So you can look at different drug likeness models, the Lipinski rule, the number of evaluations, and the Lipinski rule if it passed, the number of evaluations for Gauss rule, then if it passed Gauss rule, the Weber rule, good, the Egan rule. So you can use this as your drug likeness models for your heat compounds. For your heat compound, you can come to Empath and check that. All right. Then you can see ADMET too. Though we'll come to ADMET screening, that is pharmacokinetics. So you can still get ADMET properties from Empath. This is the bioavailability score, the solubility class, and other necessary information. And here is showing you the database where these data were created from. It's showing Swiss ADMET. So I just want to take you around this. The descriptors, you can also get chemical descriptors. Too many descriptors here.
I'm unable to see your screen.
Can anyone see my screen?
Yes, you can see.
Yes, we
Okay,
can see your
that means
screen.
it will be
Yeah.
so that means it will be a network from your end. Okay. That would be network from his end. I think my screen is, yeah, my screen is still showing. So, all right. Now this is chemical descriptors. You can show the first 100 chemical descriptors. Where you will need this part is when you are doing machine learning. Or machine learning is even too much. When you are doing a QSAR, because in QSAR, we work based on the chemical descriptors. Even when you are doing ligand-based docking, I think it will be part of. I don't know if we shall look at it, but if you are doing ligand-based docking, then you need these chemical descriptors. There are too many. Chemical descriptors are properties that describes the compound. Most of them are structural properties because every property of a compound is as a result of its structure. So you can get a lot of the descriptors, the number of acid, A log P, A log P2, AMR, and their meaning. The meaning of those descriptors. You can come to this place and pick a lot of descriptors you may need in your work. And I really give kudos to the Indians for such work. If we can have this in Nigeria, it will be very good for us. So there are too many, about 1,875 chemical descriptors for quercetin, well created here. So, all right. Then predicted human target proteins. Here also, you can derive some of the target proteins. Where this will be useful is when you are doing network pharmacology. I believe some of us must have heard about that, or we have idea of what it is. Because in network pharmacology, you do not know. Normally, most of us that have done docking, what we do in docking is we search literatures that has reported. Maybe I want to do a docking on ulcer. I begin to search literatures that have reported some targets, some ulcer targets, or we go to protein data bank and begin to look out for some receptors for ulcer. But that is actually a wrong approach when it comes to drug design. The best way to get your proteins is through network pharmacology. And in that network pharmacology, you have to create the gene of your compound, the target genes of your compound. So you must identify those genes that my compound has the ability of interacting with, or those genes that my compound can attack, either by activating them or by inhibiting those genes. So you
Sorry,
can use impact.
I have question.
Okay, yeah, go ahead.
Is it the gene or gene products? Can the drug, okay, for instance, fluoroquinolone can target DNA. Is it the gene or gene product?
The gene or gene product. I'm not getting it clear.
Now, we are talking about targets.
Yes.
So, and you talk at the DNA level. My question now is
Yeah.
that is it the gene or gene product that our drug is targeting?
Okay. Let me just explain that. You know, when I was privileged to mentor some undergraduate students. I told them that what controls our body is our gene. Our behavior is our gene. If you are healthy, is your gene. If you are sick, is your gene. So to every disease, there are genes responsible, variant genes responsible. Let me use, for instance, cancer now. If I want to target cancer, I'll have to look for those over-secreted cancer genes. Those cancerous genes. They are genes that we are responsible for that cancer. So if you are treating cancer, you're not just that cancer is not like there is cancer, but what you are treating is the genes, the cancerous cells. In that cancerous cells, there are genes that are responsible for that. So when you take drugs, you are targeting those genes. That is actually the essence. Now, these genes, these genes, you can model the genes to form protein. So when we talk about proteins are amino acid sequence. Yeah, I know that when you do your molecular characteristics, when you do your genomic studies, at the end, you expect to have some amino acid sequence from your gene. So those amino acid sequence are what you now model to form protein. So you are not targeting directly now those genes now, but those genes has been used to form the protein. So if you want to treat a disease, you look out for what are the underexpressed or overexpressed gene. Whenever a gene is underexpressed, it will cause a disorder. And whenever a gene is overexpressed, it also causes a disorder. So you have to target those genes that were overexpressed or underexpressed, either by activating them or by inhibiting them. So if I have an overexpressed gene, that means I don't want those genes to be produced excessively. I don't want that overexpression. So I will look for a drug that will act as an inhibitor. So if I have an underexpressed gene, I will look for a drug that will activate the enzymes that will help boost that gene. Now, if you have a disease, so let me give you another illustration. If you have a wound sample, if you have a wound, an injury, and you want to target those wound pathogens, you have to take samples of that wound and conduct molecular characterization. You do your DNA extractions, then you obtain the genes, then amino acid, then model the protein. So in disease, you are attacking genes. Even if you want to put it gene products, the products from the word, if you say gene product, it was actually from the gene. So if you're attacking the product, you are equally attacking the gene that produced the product you are attacking. So you can use impacts to get those genes that your compound has the ability to attack. How we do it is we look for, maybe I want to work on quercetin now, and I want to do a work on diabetes. So I will look for, in network macrology, I will look for those genes that quercetin can attack, either by inhibiting or by activating. I don't know. I will just search all the quercetin gene, just like what is happening now in this quercetin. You can see the gene symbol. All these things are gene symbols, and you can just extract all these genes. You don't really know if these genes are for diabetes or not, but your interest was just on diabetes. So you have to obtain all the genes that quercetin can attack. Then you will now obtain the genes that are expressed in diabetes. Then do a common search. You will now check, okay, I have 100 genes that my quercetin has the ability to attack, and then I have 150 genes that are peculiar for diabetes. So you will do an intersection studies using your Venn diagram tools to check if some of these my diabetes genes are they found in the quercetin genes. So you only concentrate on the common genes. So those common genes means that there are some genes that quercetin can attack, and those genes are also expressed in diabetes. Now, if you do that common search and there is no common gene, it means that quercetin will not have the ability of attacking diabetes gene, because there is no diabetes gene found in quercetin. So by the time you have a common genes, maybe out of all the genes, 10 genes which were expressed in diabetes were also found in quercetin. So you will now only narrow your work to those 10 genes. So that is the essence where I said you can use MPAT to also get the predicted target proteins, but we call them genes because they are gene codes. And that is why if you are doing network macrology, you now get to a point where we'll do gene ontology. You will do gene ontology, where you can use David's or Chinese Go for that. But though this training will not cover network macrology, but we have another training for that. So when that comes, you can leverage that opportunity for network macrology. It's very important in drug design. All right, are we together?
Sorry, I've not explained that gene ontology. What is gene ontology?
Wait, let's just leave that so that let's come back to what we are doing. Should I still continue on the gene ontology?
No, sir.
Yeah, so let's just leave that part. When the time for that training comes up, so you can avail ourselves the opportunity. So we just have to continue with the module for this training, or maybe you can private chat me so I can give you exposure on that. Thank you so much. So these are what you can do with MPAT. MPAT has a lot of features. These are what you can do with MPAT. You can search by plant, you can search by phytochemical. Then lastly, you can search by disease. You can search by disease, which is the therapeutic use. So you can search by the therapeutic use. You can come down here, but this is not really necessary for us. Here is if you are interested in a plant. Let's say I'm so much interested in Comolena odorata. Maybe that was because it's my first the plant I started working on. So I can if I don't know the possible disease that I can treat with Comolena odorata, I can use MPAT to search for those diseases that Comolena odorata can be used to control. Those diseases I can treat with this plant. So you cannot do a lot of research on them. You can also do this by using ethnobotany. So what you just do is to come here, search by plant. So if you are searching by plant, it means you want to search the disease associated to your plant. But if you are searching by disease, it means you want to, maybe I choose let me choose abortion. No, okay, acquired. Let me use ACE for instance now. So if I want to do a work on ACE, I search by disease. So what it will do is that it will provide some of those plants. You will see some of those plants that has reports. Some of those plants that has been used in treating or trying to control ACE. So this will enable you. Maybe you are doing a work on let's say you are doing a work on stroke. You can just come to this MPAT, search stroke. Then it will provide some of the medicinal plants that has the potential for treating stroke. So here you have seen Abrus precatoris, you have seen Abza amara, you have seen aloe vera, asparagus, sumo. So these are some of the medicinal plants that has a little evidence for AIDS. So it is very, very important. There are about 13. So you can show all by saying, okay, show all the entries 100. So where this will be useful for you is if you are designing a work on a disease, then you are looking for plants that you can use. And if you watch, these are the sources where those reports we are gotten. So these reports we are not just gotten by prediction, but by proof, experimental studies. So you can search by disease, and you will see the plants that Camellia sinensis don't work on this. That is a green tea. So Carica papaya that you can cure is okay. So you can explore on this and do a work on that also. So here we searched by disease. We searched by disease. That is this place. While here is search by plant. You can click and choose the plant you want to work on. Then you want to find those diseases that that plant can be able to treat. So this is Cumulena odorata. You can be able to see the disease that are associated with those common diseases that you can use the plant to treat. So let's see all. Let's see all. So here we have insecticides. So Cumulena odorata has the ability to be used as an insecticide, parasitopatholysis, wounds and injuries, antibacterial agent, antifungal agent, anti-infective agent, so and so on and so forth. So you can now do a work on each of these diseases just for Cumulena odorata. So this is why we call it phytoinformatics, getting information as much as possible related to your plant. Related to your plant. So thank God for such kind of database. Right. So I think at this point I'm done with impacts. So do you have a question to ask for impacts? Then I move to another database. Can you hear me? Are we together? Do you have questions?
Good evening, sir.
Yeah, good evening.
Are you there? Good
Yeah, I'm
evening.
with you. I'm
Okay,
with you.
so I don't want to, be clarified. So what I do is just go to my browser and type impact. And once the website
Yes,
comes up, I can now instruct it, give me any information I want.
yes. You just come to your browser and type impact. Then once you type impact, let me show you. Can you see my screen? Can you see my
Yes, sir.
screen? Okay, so I'm typing impact.
No,
Impact
you are up now.
database.
I can't see it now.
Oh,
The screen is on. The screen is on. No.
yeah,
Hello,
that
sir.
should
I
be.
can't see it now.
So sorry,
Yeah,
it's
but
network
I'm
from
not
your
seeing
end.
it. Wow.
Nigeria network. That's a problem. Yeah.
Okay, we
So
can see your screen.
once you type impact now, impact will open like this. Once you type impact and choose the first option, impact, it's open like this. You go to browse. Once you click on browse now, so what do you want to browse? Do you want to browse phytochemicals based on plant or based on the chemical? This one is based on class, but we don't need this part.
Anyone at
Then
all.
yes, anyone you can now work on, do a work, depending on what you want to do. Then do you want to search by disease? Disease based on plant or disease, and then you now get those plants that are used for that disease. That is the second option. But the first option is the plant. Then it will give you the disease that the plant can be used for. So it's a very nice one. But whenever you use impact, please make sure to cite impact. Make sure you cite impact whenever you use impact in your study.
Okay, sir.
Yeah. At least
Very much.
let their metrics too be reading, as we are all hoping to have a better H index. Let their own H index be
Yes, sir.
going. Yeah. The academics, I don't know what is doing. I don't know the problem with H index, but that is by the way, Sha. We all want to have a high H index. God will help us. All right. Do you have any other question for impact? Okay. So the silence means no. Then another database is Dr. Dupes. This one is created by America. It's owned by America. So you type Dr. Dupes. Dr. Dupes database. So once you type Dr. Dupes database, then it will
because
open.
you can't see your.
You can't see my screen?
Yes.
Can anyone see the screen?
Yes, we can see it.
Yes,
Yes,
I
we
can see now.
Yes,
All
I
right.
can see your screen.
Oh, thank you. So let us move together now. If you are with your system, you can just type Dr. Dupes. You see the first option, Dr. Dupes database. So it's also another database, but not as impressive as we have in Impact. I rate Impact as the #1 database for now. Yeah, for now. That is why I have to start with Impact and took my time to play around Impact. So Dr. Dupes is it's owned by U.S. Department of Agriculture. I just switch. What? By the year ending, or probably next year, our Nigerian database will be out, so we can also enjoy our own.
Amen.
Yes, my
Sorry,
team have been doing
you mentioned the name of that database. What is the name of that?
the
Dr.
Nigerian own.
Dukes.
No, as in Nigeria.
This
What
one,
we are anticipating.
leave that one. That one, it was empty. I don't know what they did there.
You mentioned it
That
last
is
week.
just type. You can type Nigerian phytochemical database, and it will pop out.
Okay.
They took grant and did that, so that was the problem. Nigeria phytochemical database. It was money from grant. They used to show the federal government that look at the 50,000 you gave us. We have done something from it. So that is it. NMPPDB. Okay. So coming back to Dr. Dupes now. How do I use Dr. Dupes? It's very simple. Once you have opened Dr. Dupes database, you come to search. This search. Filter by type. You leave it in plant. Choose plants. Choose plants. Because we are searching by plants. So you choose plants. Then type the name of the compound you want to search. Maybe garlic. I want to search garlic. That is a allium sativum. Okay. So the first thing is you select the entering type. So I want plant. Then you don't only for plants. You can use that for. You can get biological activity. You can get chemical information, syndrome, ethnobotanical activity, and so on. So, but we are looking at getting bioactive compounds from the plant. So you choose plant. Then when you choose plant, what plant do you want to search? If I continue with my Cumulena odorata, so most times the plant you may want to search may not be found in some of the database. You don't blame yourself now. It's not automatic. It's the one that they were able to access that they deposited. So I think my plant now, my lovely plant is not here. Cumulena odorata is not in Dr. Dudes. So I will enjoy the one I have in Impact. But if your plant happens to be found in all the database, keep downloading each of them. So that will form the assignment for today. So let me type garlic. I want to search garlic. Once you type garlic, you will see the scientific name, Allium sativa. You click on garlic. Once you click on the Allium sativa, it will open you up to the database for garlic. So you're going to see the bioactive compounds found in garlic and the parts, the plant parts. The plant parts. So you can still remember what I told you, that you download based on plant parts. So these are some compounds with the plant part. You have a lot of them. You have a lot of them to do here. You can go up to next. So there are too many. You keep downloading. The funny part is that it must be downloaded one by one. That is just the funny part, and that is where the stress is. Where the stress is in downloading these bioactive compounds one after the other. But like I said last week, if you can do it once, then you have already built your own database. So you will not be going again to download, except you are working on another plant. But for those of us who are in academics, I will advise you, if you are doing a research, concentrate, just be specific until you hit at a point. You know, sometimes I was sharing, I was addressing some researchers. I was asking them, every year you have project students. How do you design your project? You just come this year and choose any project, give to the student. Next year, you choose any projects and give to the student. But you have a plan, a goal. So your research is now based on goal. That this year, this student will be doing this part, and next year set will continue the job, this part, until I hit my target. So you can develop, you can say, okay, I'm interested in moringa. So you have 10 students, they're going to work, do different parts, different work on moringa. So you concentrate until you hit what you want in moringa. So once you have developed a moringa database now, it becomes easier for you to work, do different disease on moringa. You can even do docking about, there's a work we are doing since last year. For each plant, we are doing a docking work about we are picking 150 disease for each plant. So we want to also create that disease database for each plant. So when you pick a plant now, we are doing in silico studies for about 150 disease for each plant. So you see that we just built a database for moringa and begin to do so many work with that database until we hit the best result for moringa. Then we can develop a product for it. So I will advise you to just manage that stress in downloading these bioactive compounds. Once you do that, once you have done that forever, it will be stressful, but we must do it. We must do it. So you download, you copy, and go to your PubChem and download in PubChem. Then you can click on any of them to get other informations you may need. But I think there is no much useful information in this Dr. Deuce, apart from getting just the bioactive compounds. It is not as rich as impact. It's not as rich as impact. Okay. So you have all. You have all based on the plant also, based on the parts, the plant parts. Very important, based on the plant parts. So another thing you can do with Dr. Dukes, if you don't want to search by plants, you can search by biological activity. You can choose biological activity, then you search. So here I want to look for the biological activity of garlic, because here I was searching based on their chemical data. So let me quickly search biological activity. Let me see something. Let me say diabetes. So now I'm searching based on biological activity. So before it was based on plants, so you search plants. But now I want to search some biological activity similar to what we did last in impact, where we have the therapeutic use. So I can search diabetic, anti-diabetic, and you have results from that. You can also have results. So you can see some of the compounds that are anti-diabetic. You can use here to see some of those plants that are anti-diabetic. So you can use this. But then here is not showing the plant where these compounds are coming from. You're just seeing the list of compounds that possesses anti-diabetic potentials. So probably if you click on each of them now, you can now get the source of those compounds. You can get the source of those compounds. But I don't use Dr. Dukes for any other purpose except just getting the bioactive compound, because the other information it has, they are just scarce. It's not enriched enough. So I always use it if I'm getting bioactive compounds. So we have seen two databases now to exploit bioactive compounds. Right. We have a question for Dr. Dukes. Then I introduce another database. So you always remember to cite them. Cite them when you use them. Always cite them. You can always cite them. You can check about. You cite them. And in publications, in your materials and method, you always state the dates. If you retrieve some compounds, from this database, you must state the dates. The date you retrieve those compounds from that database, you must state the date. It's important. If you see some publications, you'll be able to see what I'm talking about. So you must cite impact. Dr. Dukes, if you make use of Dr. Dukes, you must cite Dr. Dukes. It's important. Please, if your mic is on, mute the mic. I'm not hearing myself clearly. Okay, so we have established that for Dr. Dukes. So the assignment you have today now is continuation of the previous assignments, but that previous one was based on literature. So after today now, you extend your search, your download now based on database. So, but I will advise you to use the plant that you are going to work on in your project. So you use the plant you're going to work on your project, so that it will not be after the training. You start looking for what project am I going to do. So you can just define the work you want to do now and begin to search the plants. So that at the end, you already have the data you need for that project work. So it will not take time again. Do you have any question? For
Hello, yeah,
Dr.
I
Dukes.
have a question. I have a question.
That assignment is not clear.
Okay, sir, please, I just
Yeah.
need clarification because I joined a bit late. Now, what I do is once I open the database, if I'm searching by plants, I will look for the plants and then search for, if I'm searching for the compounds, I'll check for compounds. I'll highlight the name of the compound and then go to PubChem to search for it and download. Abi?
Good. Yeah,
Okay.
yes. Yeah.
Okay.
So you can imagine if you are doing
Thank
that for 2,000 compounds. But please, why doing that?
Okay.
Keep glucose beside you. Keep glucose beside you. Why doing that? Keep glucose and keep water.
Okay. Okay, sir.
Thank you.
Thank you.
I don't quite.
All right.
Just
We have seen one database owned by India. We have seen the one by America. Let's see the one by China. We have another database, TCMSP. TCMSP. So just type TCMSP database. All these databases are phytochemical database. TCMSP database. Okay. So once you search TCMSP database, you pick the first one. Don't pick this one that has common. So pick the first one. So I believe we have some people who can interpret China here, right? You can interpret the Chinese language. So here is your area now. Okay, can you see my screen? Can you see my screen?
Yes, we are calling you.
Yes.
All right. Okay, what I did was I just changed the translation to English. So you can do that from here. Yeah. You can translate it, change the translator to English.
how did you?
Now, the issue we have here is most of the plants in China are not dominant in other parts of the country. So if you may not find your plant here, just few of them that you can find here. Let's say I want to look for garlic. Most times you may search and you may not find. Yeah. Do you want to ask a question?
How did you change the Chinese language to
Okay.
English?
So it has even come back again back to China to China. Look at my cursor. Can you see my cursor? Beside this star, up, your top right corner, top right. You see my cursor?
Yes, I can see it.
Before the star. So if you click on it, you say language Chinese simplified. Click English. If you click English, then it will turn to English. All right. So we are searching based on herbal name, chemical name, based on inch key, target name, based on disease name. So you can search by disease, you can search by target, you can search for target here, you can search for disease, you can search by the name of the herb. The name of the herb. So most times, if your herb is not a Chinese herb, if the herb is not a Chinese herb, I don't know. So, but let me just use the example here they gave. Citrus. If you want to search for citrus, any of the citrus, you search. So you can see citrus rantum or reticulata. But most times the plants you may search for may not be found here. But if you are lucky that the plants you are working on is found in TCMSP, good for you also. So once you click on it, the one you want to search, you scroll down. This place is scanty as well. Okay. All right. So if you look at it now, you are seeing name of molecule. Here is first ingredient. You can see some of the compounds. Some of the compounds in citrus reticulata. So if you are lucky that your plant is found in MPAT, Dr. Dukes, and this China database, the Chinese database, by so doing you'll be achieving more than the 1,500. Although there might be repetition, but you are going to clean the data to remove repetition. So the same way too, you copy from here, you go to PubChem and you download one after the other. In fact, this database provides other informations on the phytochemical parameters, and it also provides information on the some of the macrokinetics parameters. I can see CACO2 for absorption. I can see BBP, blood-brain barrier. I can see DL. So you can exploit this. It has about 63 entries. So there are about 63 compounds for this citrus reticulata. All right, so there's nothing really much. There's no much deal in TCMSP. But then you can also use this to check for related targets. Here is ingredients. The ingredient is telling you the bioactive compound. You can search related targets. Once you click on related targets, you can also get some of the targets. Some of the targets for your compounds. Like you can see 5-isopropyl 2-methyl bicyclo 310H2A. All of these target this gamma aminobutyric acid receptor subunit alpha 1. Why this one target cytochrome P450 cam? So these are target sourced from drug bank. Now I will give you don't know, but I believe most of us here are staffs, right, in different institutions. So I will encourage you to create account with Blood Bank. Register in Blood Bank. Yes, register in Blood Bank, because most times if you are accepted in Blood Bank, you have access to their drugs. You can use that for drug recovery. You can retrieve all the drugs that have been approved for cancer, then use them for your work. It's very important. I will encourage to do that. Go to broadband online and figure it out. Try and just register with them. They will give you approval so you can have access to the drugs in broadband. It's very important in drug design. Though it's not in macroplasy, it's for drug purpose, but you can register in broadband. It's important. You can only do that with your email, your institutional email. That is only what they accept. They don't accept personal Gmail. So you can get disease targets. Here is on network. The network, of course, network I always built using Cytoscape. So you can't open now. We should expose you to the use of Cytoscape in network pharmacology when that training comes. Yeah, this is target information. This is network parameters. That is, by the way. You can search by related disease. Still under your plant, some disease that plant can treat. So here we are seeing cardiac arrhythmias, eczema disease. So all these are useful for you. It all depends on what you want to do. All depends on what you want to do. They are useful. They are useful for you. That is that. That's when you search by the name of the herb. You can search by chemical name. So which chemical do you want to work on? Let's say this hydroquinone that is here. You want to get information about hydroquinone. That TCMS can also give you that information about hydroquinone. And one thing about I love here is that you can get derivatives of hydroquinone. You can get 56 analogs of hydroquinone from here. Then you download them one by one. So you can search by chemical name. You can search by disease name. So you can search by disease name. Whichever one you want to do. Oh, the malaria is not that disease. So on your own, just look around this database and see what you can do out of it. So what you can do from there. All right, that is for the Chinese database. The fourth database that we're exposed to is phytochemical interaction database. So type phytochemical interaction database. On your browser, search phytochemical interaction database. All right. You pick the first one.
Are we on
It
the
seems
new
it's
database?
Yes.
Hello? Phytochemical?
Yes. Phytochemical interaction database. That is the name. PCID. Phytochemical interaction database. It's also owned by China. So once you type phytochemical interaction database, the first option that comes out, PCIDB. You click on the PCIDB. This one too is dry. No much. There's no much useful information here, but you can still make use of it also. if your plant will be found there.
Can anybody hear me
What I do is that I don't underestimate
from my hand? Hello?
uh any. I can hear you. Look, can you hear me?
Yes, I can hear you now. I want to be sure if it's not from my end.
Okay. So what I do is that I okay. What I do is that I don't underestimate any database. So some of you might say, okay, all of these ones now they don't have useful information. So let me leave them. Even if you can get 2 or 3 bioactive compounds from there, it's important. It is important. It's very important. So this is phytochemical interaction database. What you just do is to come search by species, go to this species and type the name you want to search. Search by species. The example here is Solanium. That is garden egg. So once you type by species, if you are lucky to have results, fine. If there is no results, fine. So when you type by species, you come down to metabolites. You come down to okay, let's specify the species we want. Maybe let's just pick any of them. Solanium. Okay, the first one. Just let's pick this genus we want. So as you pick the one you are interested in that species, you scroll down to metabolites. So there are 18 metabolites here, which are the bioactive compounds. So there are 18 bioactive compounds. So you also look at them here. Abutyloside. So that is wonderful. Abutyloside A, abutyloside B. Oh, so you mine them, you copy them and download. You can also get the proteins, the gene of those of the plant, the genes that the plant has the ability to attack. So you can also get them from here. You can also get related disease. But that is by the way. Major interest is on the bioactive compounds, which are the metabolites here. So you can get the metabolites. So when you get them, you join with your compounds, and they are increasing. Yeah, you see, you find that there will be that the number of compounds will be increasing. At the same time, you can equally search for a plant here, and the plants will not be found in the database. So you move ahead to other database. All right. So this ones, they are not much common.
Okay.
Not
I want to take,
not really.
when we search by species, which
Yeah,
species did we pick?
I picked Solanium. There, okay, let me go back so that you will really get it. Let me come back to species. Okay, or let me just go back entirely. Okay, so search by species. You can pick moringa. You type moringa. So once you type moringa, if moringa is found,
Okay. Am I
it will
going to
bring
pick
up
plant
all
family?
the. No. Just come to this search. This search by species. Leave all those ID and name and leave the right hand part. This left hand part is for metabolites. So on this species, you type. I just type moringa now, and I search. So once you search, if the database has information on Moringa, it will come up like this. Then you are seeing Moringa oleifera. So I want Moringa oleifera. So I click on the Moringa oleifera. So Moringa here has 9 metabolites. This Moringa oleifera now has 9 metabolites. Moringa peregrina has 6 metabolites, while Moringa pteridgiosperma has 1 metabolite, according to their database, based on what they were able to deposit here. So you click on Moringa oleifera, it will open up the dashboard for the Moringa oleifera. Then you scroll down, you will see metabolites. Once you scroll down, you see metabolites. So there are 9 metabolites. There are 9 metabolites. So that is it. There are 9 metabolites. So when you just scroll down, you have seen metabolite. Then you begin to copy them and download. So here you are seeing Ramnetin slash.
We are just need to look.
Can anyone see the screen?
Yes. Yes.
All right. So here in the first name, you are seeing Ramnetin slash 7-methoxyquercetin. So they are the same name. You can decide to only copy the Ramnetin and download, or you copy the 7-methoxyquercetin, or you copy 3,5,3,4 tetrahydroxy 7-methoxyflavone. So they are all the same thing. So you don't copy everything here, just pick one. So those slash are they are the synonyms. So it's a good thing for. So this is how many now? MPATS, Dr. Durs, the Chinese database, and the other Chinese database, the PCIDB. That is 4 database. Then lastly, let's look at coconuts. Let's check coconuts. Yeah, somebody mentioned coconut, that you can download all once from coconuts. That was last week. I said I'm going to search, confirm that information. So let's look at it. Coconut database. So I think yeah, a coconut doesn't really mean that we are mining from coconut plants. It's just an acronym for the database. So coconut database. Coconut database. That is an interesting one. So you click on coconut database. That is coconut database.
Please, sir, which country has coconut database?
Okay, I don't think if it's a country is let me scroll down. Is an organization. It not is a collective effort.
Okay, sir.
It's not owned by like for instance now, if we succeed in our own now, I don't think if I even call it Nigeria database. If not, we'll say it is owned by Nigeria.
Yes.
But I just want it to be named after. I just want it to be named after Nigeria. So I learned that some of us, our concept note has been approved by NRF. Congratulations to those people. For those that their concept notes was approved by NRF. So you are hoping towards collecting from 30 to 50 million again. Make sure as you collect the money, do what you are proposed to do. As well. So this is coconuts. Coconuts. Ake. Coconuts. You can see how they form the name from collection to C. Open. Oh, that is cocoa. Then natural. These people are very wise. This is their own acronym, choke. So now, what do we do here? You can search by your molecule. You can search by your organism. The organism here is your plant. So let me move to what we really have, which is a search by the plant, by the organism. So type the organism you want, the name you want. Let's say Agerentum conizoid. Agerentum conizoids. You search. Once you search Agerentum, you have, if Agerentum is found in their database, then you are going to also see some of the phytochemicals found in Agerentum. Oh, so there's about how many? Okay, about 116. So you see, only this coconut is giving me 116 for Agerentum. Let me see impact, if impact has Agerentum, so that I will surprise some of you. Yes, because most times now people will just come and say, so my plant is not found in any of the database. So this is Agerentum for impact. Then you see what I'm trying to explain. So you can imagine that if you now combine your own wet lab. So look at this. Now, how many are you seeing here now? Can you see the entries? How many entries below?
513.
513.
613.
So impact is giving impact is giving 513 plus coconut giving us 116. You have gotten enough, except reputations. Join with your 50 database, your 50 literature reviews. We are still giving room for reputation. You are getting enough. So some of you are beginning to be afraid, but please don't be afraid. So you begin to copy the names. This is better. Okay, there's no name for this. Is 5041. The others, I'm saying their names properly. GG. Okay. Let me see how I can figure out this one. The one without name. This one without name. Let's see how we can get the name. This 504 cannot be the name. Okay, let me click on this because we will need the name to go to PubChem. Now, it is also possible that you might take the compound to PubChem and it's not found in PubChem. Do you think so?
It's possible.
Yes, many of them, sir.
Many of them. That is why the training we had last week exposed us to modeling. So you can now find the structure of that compound. Because I told us, I mentioned that last week, that most times you may try to download the structure and you see that that structure is not found in this database. So some of us might say to throw it away. You can now model it. But let me take for instance that you got a compound in impact. Then you went to, you can download from impact, but it will not be the compound CID, the normal CID for PubChem. It will carry the ID of impact. So if thank God this idea came now. If you pick a compound from impact to PubChem to download and is not found in PubChem, the only reason why I'm taking us to PubChem is for uniformity. So that all your download will bear one CID. But if you have taken a compound from impact to PubChem and that structure is not found in PubChem, download it in that impact. So you can download it from impact. You can download the 3D from impact. So let it be that one was not found in PubChem, because the ID, the ID does not have any role to play in your docking. The ID is just their identification. So we can still identify the compound from their ID, because some of you might encounter that issue where you pick some compound to PubChem and they're unable to, you cannot see them. Even too, you might see a compound from your literature and you copy the name from the literatures to your PubChem and you could not find them. That becomes a problem. For that of impact, it is not a problem because you can download from impact. But the one you will notice from your literature. So what you will do is that you will not give yourself extra stress by looking for, please don't overlook those compounds that are not found in PubChem. We need all of them. So if you encounter such, look for the structure of that compound and draw the structure, draw it using ChemDraw or ChemSketch. We did that during our free training last week. The free training we organized on macro modeling. Maybe some of us, we are not privileged to be part of that training. We are having another free training next week. Of course, the free training is not interfering with this training. No, we'll also take let's pause and go for free training. The free training comes up next week, Wednesday and Thursday. That is on molecular modeling. So I will share the flyer with us here. So if you have not registered for that training, it's free for 2 days. Molecular modeling. How we advise you to also register. It will still help you. Then tomorrow by 8 p.m. is our webinar. We're having a webinar discussion tomorrow by 8 p.m. I will be glad to see all of us tomorrow by 8 p.m. for that webinar session. We are discussing on feature-based drug design. It is still part of what you need it. You need it in this macrognosy, because like somebody asked me in my DM that I'm so much interested in 150 bioactive compounds. Will I dock these 150 bioactive compounds? The answer is no. We are not going to dock all these 150 bioactive compounds or the 1,000 bioactive compounds or the 1,000. In fact, we are not going to dock all of them, but we are going to now screen them and select the ones that has the drug-like features. Imagine among your compound there is benzene and there is toluene, xylene, methylxylene, all those benzene, some of those benzene-like compounds. that does not have any drug-like features, you do want to stress yourself and start docking them, considering the time a docking will take. So when we get to natural product mining, we shall now explain to you how you can filter your bioactive compounds and select the ones that are drug-like. That is why we kept that module for natural product mining. So in tomorrow's webinar, we are discussing the role of this feature base. Of course, like I always say that whatever is a compound can do is as a result of the structure. So what kind of features are found in a chemical structure that will enable it to be an effective drug? That is the aim tomorrow. What kind of feature, the role of features, structural features, their role in a drug-like compound. And that lecture will be delivered by hello? So that lecture, yeah, I will send it, yes. That lecture will be delivered by one of us. By one of us here, and I so much respect him. So that one now is discussion. So we'll come and talk. We may even talk and forget that time has gone. So thank you for that. So I was trying to see how we can get the name of this compound that we discovered here. Maybe I've not been able to get that name. But you can copy, let's see if you can copy the SMILE code, any of the identity to PubChem. So if we can lay hands on the identity, then we can take it to SMILE code. You can even download from here directly, but it's in 2D. It's in 2D. So I'm looking for, okay, this is the IUPAC name. So this is the IUPAC name. Good. So anyone that does not have the name, you can open them up and pick up the name. So that means after you download, you rename, because you cannot keep the name with this. You rename to this. But at the same time, whatever name they bear doesn't matter. You don't need to know them. Why downloading? We're only interested in get the compounds. We don't need to know their names. But the reason why you need the name now is so you can copy this name to PubChem to download, because you can't copy this code to PubChem. So you need to copy the name to PubChem to download. All right, so Coconut here gives you 116 data, 116 bioactive compounds for Agerantum conizoides. Very, very wonderful. All right. You can go to it. Please mute your mic. Mute your mic. So you can search by molecule. Is there any molecule of interest? Maybe quinone. You can search by molecule. But those ones are not our interest. But you can also search by molecule. Now, where this is useful, where searching by molecule is useful, like I explained before, is where you want to get derivatives. You want to get, like I typed quinone for searching by molecules. I typed quinone, and then I'm saying 1,653 compounds that has that quinone scaffold, that has this quinone scaffold. So they must have that quinone scaffold. So if you are doing a QSAR or QSPR, then you have this. I think we also have a training for that. That training should be by we have a free training by March on Curious AR. We have a free training by March on Curious AR. So by next week, we shall publish February free training. So you can also enroll in that February free training. Then you can also enroll on the February training on the green chemistry. It's a 1 month training just immediately after we finish macrognosy. We are entering the green chemistry. It's also a 1 month training. So this one is searching for phenol. You can get the derivatives of phenol, download them one by one after the other. So that is what you can do from. I think coconut is good, is enriched. Then remember, once you use any database in your work, you must cite that database. You must cite the database. So you can get that from the citation here. You must cite that database. Then as we conclude our database, I have 2 bonus database to give to you. We have given you 5. Some persons are familiar with PhytoHub. We have another one, PhytoHub database. There are lots. I have a repository that has many of these phytochemical database. So PhytoHub database, I will just show you, but I don't use it because it's not simplified. This is PhytoHub. PhytoHub is another database, but it does not specify the plants and their bioactive compounds. That is the challenge in PhytoHub. You just see compounds from plant source. Compound from plant source. And most of these compounds are nutraceutical. They are nutraceuticals. So the compounds are nutraceuticals. So you can click on compounds, or you click on foods, so whichever one you want. So if you click on these compounds, you will just see list of compounds, but it's not based on plants now. So that is why I don't use a PhytoHub, because it does not give you room to search a plant and the bioactive compounds. So you don't search plants and the bioactive compounds. You just get information of the bioactive compounds. So here is not even room of the plant, and of course you can't be doing a work. You cannot be doing a work without getting information of the plant where that is gotten from. So here there are about how many of the plant? There are about 2,696 compounds from plant-based compound. So maybe if you are doing a work without, maybe not plant-based, not based on a
So sorry, how did you check for plant-based? Because you said it's not based on plant-based. So how did you know?
Yeah. No, PhytoHub does not search by plants. So it means you may be able to use it. It just gives you compounds that were derived from plants, but you don't search. Maybe like before, you type a plant and you get the bioactive compounds. Here you just get about 2,700 bioactive compounds, but it's not based on the plants. But if you click on each of them now, show entry, then you can see the full details of those bioactive compounds. So there's no need for you to exploit PhytoHub, right? There will be no need for you to exploit PhytoHub, except you want to get information of any compound at all, but there's no need for exploit PhytoHub. But you can search based on food, because they are nutraceuticals. So if you are looking for compounds that can be used that are nutritional and still medicinal, so you can exploit them here. Like this one now, Acrola has about 169 food ID. So if you click on it, you'll be able to see the food information, which are their nutritional benefits. So now, boy, let me just give you a hint now. Let me give you a hint. I think this will help you. If you want to use PhytoHub to search by plants, come to this food. Once you come to food, now you will see names of plants. So under the food, you will see names of plants. So we are taking this plant as foods. So you see names of plants, but the plants here actually are edible plants. All the plants here are edible plants. So you can now search, depending if your plants you're working can be found here. Maybe let me search garlic. Let me come to search and type garlic. So there is garlic. And then once you click on garlic, then you scroll down. You will now see some of the bioactive compounds found in garlic. So this is the only way you can use PhytoHub to search for plants. You go to foods. But all the plants listed in PhytoHubs are mostly nutraceutical plants. So when you go to food, you can now copy those compounds there. So you have seen the compounds, so you can copy them. They are not much, 25 compounds, and the compounds here are just dietary compounds that contribute to the proximate properties of that plant. But it's still useful. You can still get related metabolites. It's still useful for you. So that is the way you can use PhytoHub. You come to food. You go to food. You search the food you want to eat. I can try moringa. So you see, moringa is not a food. So there's nothing like moringa, because I can type turmeric if I'm working on turmeric. They are supposed to be turmeric here, but it was not identified in there. Okay, let me see, turmeric. Okay. So if you are lucky that what you're looking for is found here, you make use of it. Otherwise, you also leave it. Okay. We have one, the last one for African natural products. African natural product database. African natural product database. So this will be the last database for tonight. African Natural Products Database, AMPDB. So I think we have one now for Africa. So we always look at how it is. African Natural Product Database. Let's open it and see how that database looks like. So the backbone in plants is phytoinformatics. That is why we have to start with that module. So this is African Natural Products Database. Very, very interesting database. So how do we exploit this the database? You come to browse. Can you see my screen?
Yes.
Yes, yes.
So you come to browse and you go to species. You can select by compound, but our business, go to species. And so on that species, you have from A to Z, but you can equally search. Let me look at for, we used to search before for the plants. This is A to Z. This A to Z will be stressful for you. Let me check a place where you can easily search. You only have a place where you can type and search, except it's no more available. Because you trying to searching by alphabet, it will be stressful for you to locate the plants. Okay, except that is the only way you can.
Search is up there. You can see search up there now. Can you hear me, sir?
This
After
one?
the browse, you can see search.
Okay,
After
okay.
browse,
Yes, sorry.
Can't it go?
Yeah. Okay, so just type the plants. Let's see. Okay, let me just work with Morinda. You type. So, okay. Okay, fine. So this has provided us some of the bioactive compounds under the search Morinda, but I will be specific now. Morinda oleifera. So Morinda oleifera. So we have 16 compounds for Morinda oleifera. That is how you know African database. As well. You see why the one became may not. Well, let me just keep quiet. So there are 16 compounds in Morinda oleifera. 16 compounds here in Morinda oleifera. You can copy them, download also. They tried. It's not easy to have such a database. It's really not easy. So in fact, it's even linked with PubChem. So don't stress yourself. You just click on the PubChem CID, it takes you to PubChem. It's linked with PubChem. So you can just go to just click on this CID. I think it will open the PubChem platform, then you download. So this is better for you than copying and paste. Which these databases will be modified to be in this form. Only if such compound is found in PubChem, then they can link it. So you see, others is showing dash, meaning not in PubChem, not linked to PubChem. So you have to copy them and check also. But PubChem is not the only database we use to download. When we get to chemoinformatics now, we'll also expose it to up to 10 database for chemical structures. Now, one will say everything we are discussing is database. Yes, because the in silico studies work with data. So if there is no availability of data, then you cannot do anything in silico. Any computer-based studies work with data. And that is why you will now start learning how to create your own data. A lot of us have wasted our data. Some of us in the high institutions, if you go to your libraries, your HOD's office, you will see a lot of projects, work that have been done, submitted, so many years. What happens to those data, those results they got? We just throw them and we give them the A and they go. So I will plead that we should start learning that habit of documentation, documenting your data, because that is what we use in computational studies. That's what we use. We use those data. So help us to keep our data. So far, we have explored most of the phytochemical database we can. So we have explored the database that we can. We have 7 minutes, so let me take questions. It has been an engaging session. So let me take your questions.
Hello, sir. So for
Yeah.
the African Natural Product Database, for those compounds that are linked to the PubChem, so what we do is just to click at the number, CID number, it will bring it out. Then
Yes.
we'll now download it directly from there.
Yes. Yeah.
Okay.
Can download that. Like this one now.
Direct
The one
from
that I
there.
clicked
Okay.
has opened. Yeah. Look at the one I clicked.
Okay, perfect.
The one I clicked has
Yes.
opened. So you can just click download. You can now click download
Download.
and click your 3D conformer and you download it.
Confirm.
Yeah.
All right.
Yeah.
All right.
Yeah.
Thumbs up for Africa.
We at least Africa have tried.
Hello, sir.
Okay. Bernadine, Jennifer, I'm seeing your hand up. Yeah, Bernadine. Okay.
Good evening, sir.
Yeah, good evening.
Hello.
Yeah, I'm with you. I
Sir,
can hear
please,
you.
can I get a record? Can I get a record session of this training for today? Because I missed some part.
Okay. It is not
Sir,
a
please, I'm asking if I can get the record
yes.
section of
It
this
is not.
training for today. Hello.
Okay. Yes. Can you hear me?
Yes, sir.
It is not a plead. It is a mandate. You get it. By
Okay,
tomorrow
thank you
morning it
so
will be
much.
updated in the drive. By tomorrow morning it will be updated in the drive. The total
Okay,
drive in
all
your
right,
link,
thank you.
in your mail. Yeah.
Okay, sir, thank you.
Okay.
Thank
Victor.
you, sir. God bless you.
Victor. Okay. Thank you. Yeah, Victor, I'm okay.
Yeah, yes. Thank you very much. Thank you for the insightful lecture. So my concern is, okay, in case if you are using like 5 database, you cite all of them. Then
Yes.
my another question, another case is, what if you use one, like what is the essence of using variable ones? Why you can use one, maybe like this impact, because I think impact, why not use one and reduce the headache of going through different databases? That's my concern.
Okay. One is not in one is not enough. One is not enough since it doesn't give you all the information you need. The reason is this, in our village, it was reported that bitter leaf can cure malaria. But I don't know the exact bioactive compound in that bitter leaf that cures the malaria. So I need to lay my hands on any possible information on any possible source that has information on the bioactive compounds of bitter leaf. So if I have 10 journals that has information on the bioactive compounds of bitter leaf, I will gather those bioactive compounds. If I have 10 database that has information on the bioactive compounds of bitter leaf, I will gather them. By so doing, some of the compounds I'm even gathering might be found in my repeat, no problem. But not all of them will repeat. So probably if I have 50 from impact and 10 from Dr. Dukes, maybe 5 from the Dr. Dukes might be found in impact. And I've achieved the extra 5 that we are not found in impact. Is a plus to me, because I don't know if those other 5 are even the one that will be more potent. So I don't know. You need to gather as much as possible. It will be stressful, but gather as much as possible. Yeah, thank you. Okay, Professor Justina.
Good evening, sir.
Yeah, need the cross.
Has it gotten my attention? Good evening, sir.
Yeah, good evening. We can hear you, though it's echoing. I can hear you clearly.
Please, on the African Natural Products database, when we get to where we are downloading the 3D conformer,
Yeah.
which one do we select, sir? Because I can see save beside the SDF, save beside the JSON, save beside the
SDF.
XML, and then
Take SDF. Use SDF. SDF. Use SDF.
Hello?
Yes, choose SDF.
Only SDF.
Yes, only SDF. Anytime you're downloading, choose SDF. SDF means structural data file. Structural data file.
Okay.
And also, not all the compounds may have 3D. So if you opened a compound and it does not have 3D, then it must have 2D. Then download the 2D also. That is if it does not have 3D conformer.
It's all
Then
right, sir.
yeah, pick the 2D. Okay.
Yes, I observed that you told us that Open Babel to convert 2D to 3D. So you have not shown
Yes.
us in all your training so far how to get that.
Yes. We are taking this step by step. So we are done with the database. So when we will now enter chemoinformatics, so we will now learn how to convert those our 3D, those are 2D now to 3D using Open Babel. So it's just the classes has been arranged a step before another. So I will show you how to convert that.
Please, sir, the assignment for today's class, I didn't quite get it.
Thank you very much.
Hello, sir. Before the assignment.
Okay.
Hello. Yes, sir. I
Yeah,
want to
yeah.
ask. The other time you were lecturing us, you were talking about the use of those database to increase the number of compounds that we are going to have as our own database, whatever. My question now
Yeah.
is this: you remember day before yesterday, you told us that we should use literature to get our compounds.
Yes.
Are you now saying that we should also use database to increase the number of compounds to get?
Good. In fact, I mentioned three approach. In the first class, I mentioned three ways that we can achieve large database, large data. The first one I mentioned was the wet lab, that you do your characterization. That one I involve money. You do your characterization using different solvents. Then you characterize with GCMS and the HPLC. You have your own compounds. You keep it. You go to literature, get the compounds, join with the one you
Yes.
got from your wet lab.
Wet lab.
Go to database, join everything
Okay.
together. At the end, you are
Okay.
increasing your data. Yeah.
Okay. All right. Thank you,
Yeah.
sir.
Thank you very
The
much.
assignment now.
Okay. So the please, I want to apologize for assignments, but I will not stop because even if you are the dean or you are the DVC or vice chancellor in your school, once you are in this training, please, I'm begging you, find time to do the assignment. It will help you. The essence of this assignment is not that we are even recording it or giving you score. Of course, we are not giving you one special certificate, but for you to practice and become a bit perfect. So the last assignment we gave was only based on literatures. Today now, you are now extending to database. So the same plants you have used for literature review, for literatures, mine the bioactive compounds now with database. Still based on the plant parts, unless the data, just not all of this database reported the plant parts. The only ones that reported plant parts were impact .dudes. Then I believe others did not took note of the plant parts. So those ones without plant parts, you keep them on a different folder. So for today's class now, for today's assignment, you are creating your own a new folder now based on database. Keep that folder you have done on literature review separate. Now you are creating a new folder for database. So you create a folder for leaf, the leaf parts. Maybe now you type your plant, that same plant you have used in your literature. Type it in impact, search with impact. Once it's found in impact, it may be found or not. Once it's found in impact, you start downloading them based on the plant parts. Impact will always arrange the phytochemicals according to their plant parts. So probably you start working on the leaf. That means you create a folder now called leaf. Make sure it will be different from the one you have done before on the so don't join them together. So you create a folder now for leaf. Probably leaf database for your plants. Download all the leaf, put in that leaf folder. Then if impact has for roots, download for roots, put there. Then if you are done with impact, you search for .dudes. Once you enter .dudes, don't create a separate folder for .dudes. If you have another leaf, compounds under leaf also for .dudes, download them, join them with the same leaf you got for impact. So we are not creating folder based on the database we are using, but based on the plant parts. We don't have interest based on database, but our interest is based on plant parts. So now when you now look at the dudes. You download according to the plant parts, join them with those ones for M parts. Then the rest database does not specify the plant part. So if you also make use of those database that did not specify the plant part, that means you have another folder. You title it not specified. Plant part not specified. That will be the name of the folder. So download all those ones that the plant parts we are not specified. Put them there. Yes, put them in that folder. So that is the assignment. So the assignment now is based on database. We are using database now for this assignment. If you are not still clarified, you can ask your question for clarification.
Yes, sir. Hello, sir.
Yeah, with you.
Yes, sir. The compound that we obtain from the database now, are we to make sure that they are the one that are different from the one obtained from the literature, or we just download any one that we find?
Download the ones you find.
Okay.
Now,
All right.
yeah. When we will now make sure that they are different is when we now join everything together.
Okay.
Both maybe for leave now, the leave in literatures and the leave in database will now join them. Once you join them, the system will tell you that there is repetition. You will see,
okay.
Yeah. So we'll now delete the ones that repeated.
Okay.
That becomes easier.
All right, sir. Thank you, sir.
So, sir,
But for now, we are not joining. No, we are not joining them for now. We are just concentrating on the database. Yeah.
So, sir, we are still continuing with the plant of interest, right?
Good. Yes. Yeah. Like I said before, let the plant of interest be the one you will use in your project, so that you will not come back to your project work and begin to do afresh again. So first, we have started each step, each of the session forms part of your project. Yeah. So continue with that same plant part of interest. Don't deviate.
Okay, sir.
Yeah.
Thank you, sir.
All right. There's a person on the chat box. If you have any other question, you can do it to relate with us. Please, can you explain TCMSP? Okay, how can we cite the databases? Yeah, I showed us. You can check their about. To cite the databases, you check their about. Like this African database now, you check, maybe check their contacts. You will see the citation. If you could not see where they wrote how to cite them, then cite the link. Cite the link. But it should be okay. Somebody asked me about the African database, the people that has it. So you can see the people that has it. That is, it was done by Department of Pharmaceutical Sciences. Yeah, University of Springborg. So they have done well to name it African database. So you can check their about. They're supposed to give us their citation. Yeah, they're supposed to give us the citation. But in each case, it's just scroll to their about. Like in Coconut, there is citation. Oh, it's not still showing here. So if you checked and did not provide their citation format, then cite their link. You copy the link up and cite it. That is what you do. All right. Somebody said, how can you explain TCM? Please, I did not get the link to the Chinese database. The Chinese database is the TCMSP. The TCMSP. Look at it. That is my screen. This is my screen now. If you can see my screen, the TCMSP is this one now. Just type TCMSP. TCMSP. Maybe someone is looking for the meaning of the TCMSP, traditional Chinese medicine system pharmacology database. So that is the TCMSP. The person that asked the explanation of the TCMSP, traditional Chinese medicine system pharmacology. All right, so TCMSP database. So we have achieved a lot, and I will encourage you all to, so you pick this first one, TCMSP. So I will encourage you all to do more. We are already 12 minutes ahead of our time. So thank you very much. I believe there is no more question now. So at this point, we will meet again on Tuesday. So when we likely finish this one now, they pleaded we should give a grace for the literature for Tuesday. So that means this one will be on or before next week Friday. Hope it's okay. It is okay now. So let's put it, yeah, on or before the next session for that day the assignment was given. So you have time, you have a week. So like as we gave on Tuesday,
we have not been able to finish the first one.
They pleaded now that we should give a grace till Tuesday.
Okay.
So this one now is now a grace till next week Friday.
Oh.
The Lord is our strength. You just do what you can, but if you can for all of them too, yeah.
But again, I have a question on that. The first assignment. Actually,
okay.
This is not my area, but I'm trying to.
Yeah, what is the area? What is the area?
Actually, I'm into chemistry, but analytical chemistry. But I'm trying to switch, so it's not really easy for me. But I'm coping anyway. But I'll try. But the question I have on the first one, do you mean 50 literature as in 50 articles, then get those characterizations from each of the articles and download everything, or?
Yes. 50 articles that has information on the characterization.
Yes. Thank you very much.
Thank you, sir.
Yes, sir.
Right. Happy weekend. Enjoy your Saturday. Enjoy your
too.
Sunday. Thank you very much. We'll meet again. I'm glad to have you around. Thank you. Thank you.
Thank you so much, sir. Good night. You've done novel.
Thank
you so much, sir, for that.
you.